So hello students, I hope all of you are doing good. Welcome back to your own channel, students, Vedantu Science Olympiad. Now students, in today's class, right, we'll be focusing mainly on the plant hormones, different types of plant hormones, auxins, gibberellins, cytokinins, ethylene, abscisic acid, and how all of these different types of hormones play important role in your plant growth and development. Okay, so students, without wasting time, without even spend next 5 minutes, right, first 15 minutes we'll be focusing on this chapter's foundation, like what is growth, what is development, what is plasticity and everything. And after that, next 30 minutes we'll be focusing on the entire hormones. Okay, now first things first, students, first things first, this entire notes will be available in the description, so please download the notes from the description itself. Okay, now students, first things first, what is growth actually? What is growth? For example, if I ask you what is growth, you will generally tell me growth means increase, है ना? Growth माने increase हो रहा है, सब कुछ तो increase हो रहा है. Okay, so all of you need to know growth is nothing but increase in mass or increase in number of cell or increase in both of them. So that is called as growth with respect to humans, with respect to organisms. So growth माने increase in something. Now all of you might remember growth can be of two types. There is something called as intrinsic growth and extrinsic growth. Now what is the difference? Intrinsic means what? Intrinsic means from the inside. Growth from the inside is called as intrinsic growth. Now what is extrinsic growth? Growth from the outside is called as extrinsic growth. Now for example, humans, right, all of us, we humans, हमारे body के अंदर हम intrinsic growth दिखाते हैं. We are growing from the inside. Now one more example is, students, for extrinsic growth is, let's say you go to Kashmir this time, and let's say वहां पर snowfall हो रहा है, वहां पर क्या हो रहा है? Snowfall हो रहा है. Then what do you see? For example, this is a hill or a mountain. इस hill or mountain के ऊपर की तरफ you can see the accumulation of snow around it, right? Accumulation of snow around it. Now this type of growth is called as what? This type of growth is called as extrinsic growth. Growth from the outside it is happening. So intrinsic from the inside, extrinsic from the outside. Now one more thing which all of you need to remember is growth is generally irreversible. Now what do I mean by irreversible? Let's say आपकी height अभी, students, let's say आपकी height अभी, you are in class 6th, 7th, 8th, right? Let's say आपकी height अभी 5 feet की है और आप क्लास 12th होने के बाद आप 6 feet के हो जाओगे. So from 4 feet you or 3 feet you went to 6 feet height. Now can you go back to again 3 feet? Can you go back to 5 feet? नहीं, possible नहीं है. So that is the irreversible change. एक बार जो हो गया वो वापस उसको लेकर नहीं आ सकते आप. That is called as irreversible change. Now I'll give you one more example. Okay, I'll give you one more example. So listen to me. Okay, I'll give you one more example. Let's say I am 65 kgs right now. Let's say I am 65 kgs right now, and next year I don't follow any diet. I eat all the junk food in the world, और मेरा weight हो जाएगा 75 next year. So from 65 to 75 weight I became. Now is that growth, students? Can we call that growth? All of you, can anyone tell me in the class? Can we call that growth? 65 kg to 75 kg. Is that growth? No, that is not growth, because the 75 kg me, the future me, let's say I go to gym, I do dieting and I come back to 65 kgs. So my weight can be reduced again. So that is not growth, because that is reversible in nature. So growth is what, students? Growth is irreversible permanent change. A change, it is happening and you cannot reverse it back. That is called as growth, students. That is called as growth right now. Now let's say germination of a seed, right? Germination of a seed is also called as growth. Yes, that is also called as growth, because अगर एक बार एक पेड़ बड़ा बन गया, let's say this seed underwent germination. Now can this entire large plant again become a seed? The same seed? Not possible. This plant can produce seeds. ये seed produce कर सकता है, but this plant cannot become a seed. So that is the irreversible change. That is the irreversible change. Now I'll give you one more example. Let's say, students, I'll give you one more example. यहाँ पर मेरे पास एक पेड़ पौधा है. Okay. Now on this plant, I have a leaf here. I have a leaf here. Okay, I have a leaf here. Now this leaf expands, or this leaf shows growth right now. This leaf is showing what? This leaf is showing growth right now. Now, is this called as growth? It is a growth because this leaf cannot become a small leaf again. Jo ho gaya, so ho gaya. It cannot go back. That is called as growth. Okay. Now, students, now there are few definitions which you need to know. What is it? First thing which you need to know here is plant growth is unlimited or indeterminate. इसका मतलब क्या है? इसका मतलब है कि plants can keep on growing. For example, in the case of humans, we will stop growing height, let's say around 20 years of age or 19, 18, 21 maximum, we will stop growing. We cannot grow tall after that. But in the case of plants, that is not the case. Plants can keep on becoming larger and larger and larger, taller and taller and taller throughout their life. उनके पूरे के पूरे the life history में they can keep on becoming bigger and bigger. Okay, so they can keep on growing. Now, why is it so? What is so special about them? And why can't we grow, students? The answer is very simple. In the case of plants, they have certain types of dividing cells and they make up a tissue that is called as meristematic tissue. Primary meristem, secondary meristem. Both of these meristems help in the growth of a plant. Primary meristem, height. Secondary meristem, width. Please remember that. Okay, so primary and secondary meristem, because of these special type of tissues, right, plants can keep on growing. Okay, so primary meristem for primary growth, that is increase in height. Secondary growth because of secondary meristem. What is secondary growth? Secondary growth is increase in width. Increase in width. Okay, now students, we say that plant growth is open. इसका मतलब क्या है? The meaning of that is new cells are always added. For example, plant के अंदर हमेशा नए नए cells add होते रहते हैं, हमेशा होते रहते हैं. Okay, now plant growth is localized. इसका मतलब क्या है? इसका मतलब ये है कि the generally plant will grow only in certain areas जहां पर ये meristematic tissues are found. And this meristematic tissue generally found in the shoot and root tip. You might have heard about something called as, students, you might have heard about something called as RAM and SAM, Ram or Sham. What is this? Root apical meristem and shoot apical meristem. So wherever you find meristem in the shoot tip and root tip, usi jagah mein na there can be growth. So plant growth is localized. But in the case of animals, growth is uniform. You will never find a human with long neck. You will never find one human with one long arm and one short arm unless it is a disorder. Generally, you will not find एक पैर बहुत लंबा है, एक पैर बहुत छोटा है. You cannot find that. In animals and humans, the growth is uniform. Okay, growth is uniform. Now in the case of plant, number of organs keeps on increasing. इसका मतलब क्या है? The organs are there, right? हमारे जो organs है, heart is the organ, kidney is the organ, liver is the organ. Now they do not increase in number. They never increase. You cannot have two hearts. Not possible. But in the case of plants, the organs keeps on increasing. For example, leaf. Leaf is a photosynthetic organ and plant has a lot of leaves. हर साल नए leaf बनते रहते हैं. Plant has something called as flower. Flower is a reproductive organ. Every single season, plant will keep on producing flowers. So flowers के number increase होते रहते हैं. So plant, what happens? What? Number of organ keeps on increasing. Very important information, students. Very important information. Okay. Now, students, now when I talk about growth, I am talking about increase in living part of a cell. Yes or no, all of you? When I say growth, I am actually technically speaking about growth of a cell. जब हम growth की बारे में बात करें ना, we are talking about growth of a cell. And if you look at a cell, this is a small cell, it becomes a large cell. Now, what is actually increasing here? The increase in the living part of a cell. And the living part of a cell is called as protoplasm. So, can I actually tell, when I talk about growth, am I actually talking about protoplasmic growth? है ना? हम protoplasmic growth के बारे में बात कर रहे हैं. But there's a problem here. This protoplasmic growth cannot be measured easily. इसको measure नहीं कर सकते क्योंकि this is very tiny growth or it will difficult to quantify it, right? Very difficult to quantify it. So what do? We look for alternatives to measure the growth. For example, there is something called as we can count the increase in number of cells. We can count that increase number of cells. We can look at the increase in cell number and cell size, increase in fresh weight and dry weight. Obviously there's a difference in fresh weight and dry weight. Then increase in length, for example pollen tube increase, pollen tube growth, right? Pollen tube growth, you can visually see, you can actually see the growth of a pollen tube, we can see that. Then increase in surface area, एक leaf छोटा सा उसके बाद बड़ा leaf हो गया, so there is an increase in surface area here, that is also growth, done. Increase in volume, done. So all of these are different ways to measure the growth of a plant. Okay, now students, now what are the phases of growth? There are three main phases. There is something called as meristematic phase, elongated phase, and maturation phase. Now what is the meaning of each one of them? For example, the first one is meristematic phase. In meristematic phase, a cell is a dividing cell. It is a new formed cell. This cell has a lot of cytoplasm, dense cytoplasm, right? See here, it's the first phase. Cells are very small size, dense cytoplasm, vacuoles are absent. Obviously, this cell तो अभी अभी बना है ना, it's just dividing, it's just growing. So यहाँ पर vacuoles की जरूरत क्या है ना? There is conspicuous nucleus, cell wall is primary, and we can find the plasmodesmal connection. What is plasmodesmal connection, students? Plasmodesmal connections are cytoplasmic connection. They are the connections present between the two different cells. इसका cytoplasm और इसका cytoplasm both are connected here. Done? Done, students. Now, this is called as elongation phase. In elongation phase, a cell becomes larger here, larger. Okay. Then is maturation phase. Maturation phase, every single cell has a fixed structure and function. But generally, they will not ask you all this, so don't worry much here. Okay. What we need to be focusing, students? We need to be focusing on something called as the types of growth rates. There is something called as arithmetic growth and geometric growth. Difference क्या समझो students? What is arithmetic growth? In arithmetic growth, we have one cell. This cell will divide into two cells. When it is dividing into two cells, one cell will remain meristematic. Other cell becomes permanent cell. It will not divide anymore. Similarly, this cell again divides. One cell is meristematic, one becomes permanent. This cell divides, one is meristematic, permanent. This is called as your arithmetic growth. Now, what is geometric growth, students? In the case of geometric growth, when a cell divides, both the cells are going to be meristematic in nature. There is no permanent cell formation here. That is a basic difference between arithmetic and geometric growth. Now, students, this concept I will teach you in anatomy, because this is a power revision, right? This is a very small chapter. So this concept of differentiation, dedifferentiation, redifferentiation, I will teach you in the anatomy chapter, not here. Okay, because I want to focus on the main part of this chapter, that is your hormones. So before we go to the hormones, students, there is one more small concept called as plasticity. Now, what is plasticity, students? Plasticity concept is very simple. Let's say हमारे life में we have a path to take, right? For example, there is a path: seed germinates, juvenile phase, उसके बाद mature बन जाएगा, फिर flowering करेगा, फिर senescence and death. This is a normal pathway a plant can take. Now let's say इस pathway से ना एक plant अभी deviate हो जाएगा, you become variant, okay, technically. So everyone has a pathway in life. So if you move away from the pathway, that is called as plasticity. Okay, that is called as plasticity. That is the ability of plant to alter their development in response to the environmental conditions and different phase of life. So everyone has a pathway, plant has a pathway, it is altering its pathway, okay, based on the environmental. Let's say, I'll give you an example here only. Dekho. There is a plant called as buttercup. This buttercup plant, it will it will be different in different environment. Same plant in terrestrial condition, इसके बड़े-बड़े पत्ते होंगे, large leaf, okay? But the same plant in the aquatic condition, the leaf are very tiny or needle-like leaf. Why? Because in aquatic condition के अंदर there is a movement of water, है ना? Movement of water होगा. Now you only imagine if this plant, buttercup, in aquatic condition, if it had large leaf, तब क्या होता था? There would be a lot of resistance, ना? There would be a lot of resistance. So we don't need resistance in water condition. So very tiny leaf. This can also be called as adaptation also, which all of you know. Okay. Now students, now we come to the main topic of today's students. Okay. Now we come to the main topic of today's class, that is the plant hormones or phytohormones or PGR. What is PGR? Plant growth regulators. Plant growth regulators. Now from here onwards, the class is going to be very important. So please listen very carefully. Okay. The first 15 minutes are over. Now is a serious time. Okay. Now students, now what are phytohormones? Phytohormones or plant hormones are basically chemicals. They are also chemicals. And these chemicals control physiological activities of a plant. For example, growth of a plant, respiration of a plant, almost every single activity of a plant is controlled by these hormones. When to flower, when to divide, the cell division of a plant, everything is controlled by hormones. Okay. Similarly, in our body also, hormones play a major role. Testosterone, progesterone, estrogen, all of these sexual hormones also play a very important role, right? So all of these hormones are very important for a normal functioning of a body. So इसी तरह से plant के अंदर भी ऐसे कोई hormones हैं which are very important. Now students, now for example, all of these hormones in plant can be divided into 3 categories. There is plant growth promoters. These hormones, right, these hormones will promote the growth of a plant. There is growth inhibitor. This hormone called as ABA, also called as abscisic acid, it will inhibit a plant growth. Plant को grow करने नहीं देगा ये. Then there is a dual nature. What is dual nature? This plant is doing both. Sometimes it is promoting, sometimes it is not promoting. So these are the different types of hormones. Let's understand one by one each one of them right now in detail. Now students, please download this, because here I have given you the summary also. Summary of all different hormones, types and functions here. So this we can deal at the end, but let's go one by one from the first one called as auxin. Now auxin is a type of your indole compound. It is a type of your indole compound. Indole, all of you know chemistry, right? See, indole. This is the indole structure. So that is the chemical nature of a auxin. And precursor is tryptophan and zinc. That is, from tryptophan we can get the auxin. So in a plant, if auxin has to be prepared, agar auxin banna hai na, so plant ke paas it needs tryptophan. So this tryptophan will be converted to auxin eventually. Okay, and auxin is a Greek word to grow. So this hormone helps the plant to grow, actually grow. Now students, now auxin can be of 2 types. There is natural auxins which are present in nature as such. Then there are synthetic auxins. These auxins are prepared by humans in a laboratory. Now, what are examples of natural auxins? There is IAA, IBA are the 2 famous ones. That is indole 3 acetic acid, indole 3 butyric acid. So ये दोनों के दोनों है main natural auxins हैं. Then there is also your artificial or synthetic auxins. They are prepared in the lab. There is 2,4-D, 2,4,5, tri-D, right? So all of these are your synthetic auxins. Now here students, this 2,4-D is there, right? This 2,4-D is also used as a insecticide and a weedicide. So weedicide mainly used as a weedicide. It will help us to kill a lot of different types of dicots. That is 2,4-D. Now students, now here in auxin, let's say discovery. Who discovered auxin? Auxin was discovered by Charles Darwin and Francis Darwin, father and son. Father and son, both of them discovered the auxin hormone, right? In which plant? In canary grass they discovered here. Now, how do you discover? What was the experiment called as? It was called as phototropism. Now, what was the experiment? Let's understand. Let's say this is a canary grass here, and they noticed that this canary grass is growing towards the sunlight. They noticed that. Okay. Okay, students, we will change the timings of the classes. Don't worry. Okay, we'll change the timing of the classes. Right. So this canary grass will grow towards the sunlight, growing towards the sunlight. So they understood that Charles Darwin Francis Darwin understood that कुछ तो है इसके टिप के अंदर which is helping it to grow towards the light. Then what did they do? In one experiment, they completely cut the top part. Cut करने के बाद उनको realize हुआ the plant is not bending towards the light now. It is not growing towards the light now. Not going towards the light. One more experiment they did what? They completely covered पूरे का पूरा जो टिप है ना उसको कवर कर दिया उन्होंने. In that case also the plant did not show bending anywhere. So finally they realized कुछ तो है इस कैनेरी ग्रास या नॉर्मली एक प्लांट्स के टिप में शूट टिप में कुछ तो है. It is helping it to grow towards the sunlight. Okay, and that was next step. Next step is isolation. The isolation of auxin was done by F.W. Wendt. So F.W. Wendt did the isolation of auxin. Okay, so in which plant? Avena sativa, that is oats plant. So this you have to remember. In oats plants, they did the isolation. Discovery was in canary grass. Canary grass discovery, isolation by F.W. Wendt in Avena sativa. Now, what is the experiment called as? The experiment is called as Avena curvature test. इसमें क्या हो रहा है स्टूडेंट्स? वेरी सिंपल. इसमें उन्होंने सेम दे टुक अ ओट्स प्लांट, टॉप पार्ट को कट कर दिया. कट करने के बाद इसको हमने एक मीडियम में डाल दिया, अगार अगार के मीडियम में डाल दिया. एंड आफ्टर सम टाइम वॉट एवर हार्मोन वॉज प्रेजेंट ऑन द टिप, जो भी हार्मोन यहां पर था ना, ऑल ऑफ देम डिफ्यूज्ड इनटू द मीडियम. डिफ्यूज हो गया. Now diffuse होने के बाद we took the bar here and we placed it on different sides of a plant, different sides of a plant. And then what did we notice? Friends, what did we notice after placing the bar of your auxins? We noticed that students, जहां पर भी हमने ये बार रखा है ना, अगर सोचो हमने इस बार को अभी इस तरफ रखा है, अब क्या होगा? इस जो इस पार्ट का this side will show more growth. इस साइड ग्रोथ ज्यादा हो रहा है, इस साइड है ना, इस साइड ग्रोथ ज्यादा हो रहा है. Then what happens? If growth is more on this side, obviously this side will grow more. इस साइड का बहुत ज्यादा बढ़ जाएगा and obviously will start bending towards that side. Obviously. Okay, take one more example. Let's say this is a plant here और इस साइड पे ज्यादा ग्रोथ हो गया. Then what happens? This will keep on growing and it will bend towards the other side. So let me just explain you one more time with one more example here. Let's say this is a plant here और अगर हम इस साइड पे हम the auxin का बार डाल रहे हैं, auxin का बार डाल रहे हैं. If you are keeping auxin on this side, now what will happen? Very simple, student, very simple. That is, this side will start to grow more and more. Now here the light doesn't play a very important role also, because light can be present from any side. Now any side light is present. If light is this side also, it's better. Okay, now if auxin is on this side will keep on growing, but this side it grows less, and we can see the bending of the oats plant. Okay, because of the auxin is present on the opposite side. As students, please remember, auxin is always produced on the opposite side of the sun. Generally what happens? Okay, Ansh, I'll tell in the ma'am, don't worry. अब देखो यहाँ पर क्या हो रहा है, यहाँ से sunlight आ रही है ना, आ रही है. So this side will happen what? This side will grow more and more. It will grow more and more, and you can see the bending of the entire curvature here. Now in examination what they'll ask you, examination के अंदर आपको directly पूछेंगे what are the functions of auxin, और यहाँ पर मैंने सब कुछ दिया. देखो, root initiation, flower initiation in pineapple, it helps in parthenocarpy, that फॉर्मेशन ऑफ फ्रूट विदाउट फर्टिलाइजेशन, राइट। इट हेल्प्स इन एपिकल डोमिनेंस, मतलब क्या है, पूरा प्लांट स्ट्रेट ग्रो करेगा, प्लांट साइड से नहीं ग्रो करेंगे यहाँ पर, ओके, प्लांट्स लॉन्ग ग्रो साइड साइड, okay, plants will not grow on the sides, plants will keep on growing straight here, they'll keep on growing straight, that is called as apical dominance. What is feminizing effect, students? Feminizing effect का मतलब है यहाँ पर फीमेल फ्लावर्स ज्यादा डेवलप होंगे, okay, then there is cell division, cell enlargement. So एग्जाम में आपको डायरेक्टली ऐसे क्वेश्चन आएंगे, what are the functions of the auxin, और वरना आपको कुछ फंक्शंस देंगे और पूछेंगे कि किसका फंक्शन है ये। now यहाँ पर one thing is very important, okay, प्रिवेंशन ऑफ एब्सेशन, इसका मतलब जो एक पार्ट है ना उसको डीके होना, डीके और गिर जाना नीचे, ओके। सो अगर एक प्लांट ओल्ड हो गया, इन ओल्ड प्लांट्स इट विल नेवर प्रिवेंट, इट विल नेवर प्रिवेंट द एब्सेशन ऑफ अ प्लांट लीफ, एब्सेशन मतलब ओल्ड होके गिर जाना, ओके, लाइक डीके हो जाना। इन द यंग प्लांट इट विल प्रिवेंट, यंग प्लांट विल लाइक तू रुक जा, तेरे पास टाइम है, तू मरना मत, बट ओल्ड प्लांट के अंदर और ओल्ड पार्ट के अंदर उसको छोड़ देता है, उस वहां पर ऑक्सीजन का काम कुछ नहीं होता, ओके। सो लेट्स से वन एग्जांपल स्टूडेंट्स हियर, ओके, स्टूडेंट आंसर दे दिए इसको यहां पर, ओके। सो they can ask question like this, the phenomena which is influenced by the auxin and also played a major role in the its discovery, that is phototropism. So phototropism plays a very important role there, it helps in bending of the entire tip there, right? Now what about gibberellic acid, students? Very important here, और यहाँ वो स्टोरी बहुत अच्छा, स्टोरी अच्छा है इसका, okay. Now chemical nature is terpenes or terpenoids, precursor is called as acetyl CoA and malonic acid. Now discovery was done by Kurosawa here in plant called as rice. Now this was observed in Japan. जापान में ऑब्जर्व किया स्टूडेंट्स कि एक राइस प्लांट है जो बहुत ही लंबा है, एक राइस प्लांट है जो बहुत लंबा है, और उस राइस प्लांट के अंदर राइस ग्रेन्स ही नहीं है। स्टूडेंट सोचो, अगर हमारे पास एक लंबा सा राइस का पेड़ आ गया, पेड़ नहीं लाइक प्लांट आ गया, राइट, लंबा सा राइस का प्लांट आ गया, बहुत लंबा ये प्लांट, पर उसके अंदर ना सीड्स ही नहीं है, सो ऑब्वियसली यू आर अ फूल, राइट, यू आर अ फूल। सो दिस वाज कॉल्ड एज व्हाट? दिस वाज कॉल्ड एज बाकने डिसीज, इसका मतलब फूलिश डिसीज, क्योंकि यहां पर जो प्लांट्स हमारे पास है ना उसके अंदर सीड्स ही नहीं है, और ये बहुत वेस्ट ऑफ टाइम हो गया हमारे पास, सो बाकने डिसीज। नाउ दिस एंटायर बाकने डिसीज, राइट, it was caused by a fungus actually called as Gibberella fujikori. Gibberella fujikori is a fungus which can cause the Bakane disease. Bakane is a foolish disease. Okay, Bakane disease or the foolish disease. Okay, so isolation was done by Ubuta and Sumuki. That they can ask you. Bioassay is called as alpha amylase barley endosperm test. Now, what they can ask you in examination? What they can ask you in examination is the different functions of gibberellins. What can they do? But before we go to that functions, you need to know that more than 100 gibberellins are reported. Different types of gibberellic acids are reported, denoting from GA1, GA2, GA3 and so on. Hundreds different types of gibberellins are there. Okay, gibberellic A3 is called as gibberellic acid, and A3 was the first isolated form, okay, of gibberellic acid. Okay, they are acidic in nature. Now students, here are the functions. Here are the functions. सबसे important function क्या students? Gibberellic acid का. Gibberellic acid का सबसे important function है, it helps in the increase the length of a plant. Okay, it helps stimulate the stem growth. एक plant को ऊपर लंबा कर देगा. Now students, imagine अगर आप भी gibberellic acid को लेके ना आप भी लंबे हो सकते थे. Like imagine that condition. अगर आप 5 फुट के हो, 4 फुट के हो, है ना, और आपके ऊपर मैं थोड़ा जिबरेलिक एसिड स्प्रे कर दूंगा, मैं आपके ऊपर में थोड़ा सा जिबरेलिक एसिड स्प्रे कर दूंगा, तब क्या होगा? इमेजिन यू कुड ग्रो वेरी टॉल, मजा आ जाएगा ना, मजा आ जाएगा। बट दैट इज नॉट द केस, ओके, इट ओनली हैपेंस इन द केस ऑफ प्लांट्स, ओके, इट हैपेंस ओनली इन द केस ऑफ प्लांट्स स्टूडेंट्स, ओके, ओके। Bioassay is basically an experiment to prove the effect of a hormone. Okay, now students, now promote seed germination. अगर आप जिबरेलिक एसिड को डाल दोगे ना, if you add जिबरेलिक एसिड to a seed, वो germinate हो जाएगा, right? Promote seed germination. Bolting in rosette plants, this is your This is your NSEJS PYQ, right? Now, what is rosette of plants? First understand, students. What is rosette of plants? Rosette माने rose की तरह. For example, cauliflower, cabbage. Cauliflower and cabbage are called as rosette plants. They look like a flower, no? They look like flower. इसके अंदर अगर आपने देखा होगा, एक कॉलीफ्लावर का स्टेम बहुत छोटा सा होता है. स्टेम कभी देखा है स्टूडेंट्स आपने? कभी देखा है क्या? इफ यू लुक एट ऑब्जर्व द स्टेम ऑफ अ कॉलीफ्लावर, इतना सा होता है स्टेम, इतना सा होता है. बट इफ यू ऐड जिबरेलिक एसिड, उसका स्टेम लंबा हो सकता है. सो इट कैन शो बोल्टिंग. स्टेम बोल्ट हो जाएगा, लंबा हो जाएगा बोल्ट. ओके, डिले सेनेसेंस, मरने से, प्लांट को मरने से रोकेगा, ये ओल्ड होने से रोकेगा. ओके, दैट आर द फंक्शंस ऑफ योर जिबरेलिक एसिड स्टूडेंट्स. जिबरेलिक एसिड. नाउ इंक्रीज द लेंथ ऑफ ग्रेप स्टॉक. अगर ये ग्रेप्स इतना सा ही है तो लंबा कर देगा. इंक्रीज लेंथ ऑफ स्टेम, दस इंक्रीज द यील्ड इन शुगर केन. अगर आप सोचो, राइट, एक शुगर केन है, उस शुगर केन पे अगर आप जिबरेलिक एसिड स्प्रे करोगे, राइट, नेक्स्ट जनरेशन में उसका लेंथ बड़ा हो जाएगा. शुगर केन लेंथ विल कीप ऑन इंक्रीजिंग. वरना जब वो ग्रो कर रहा है ना, शुगर केन जब ग्रो कर रहा है, कीप स्प्रिंग जिबरेलिक एसिड. आपके पास तो शुगर केन आएगा ना, गन्ना keep on increasing in length. Okay, elongate and improve the shape of apple. Speeding up the malting process in the brewing industry. What is malting process? The fermentation process. जल्दी जल्दी कर देगा. Spraying juvenile conifers. Conifers are your gymnosperms, right? With gibberellic acid, hasten the maturity period. So ये conifers जल्दी जल्दी mature हो जाएंगे. So all of these are the different properties of gibberellic acid. The most important are these, and then we have these properties also. So you need to remember gibberellic acid क्या करता है. For that, best way to remember is a table. उस टेबल को दिखाऊंगा ना मैं, टेबल, उस टेबल का एक प्रिंट आउट निकाल दो आप. 2 रुपए लगेंगे, 2 रुपए लगेंगे आपको. 3 पेज है वहां पर. ओके, 3 रुपए ले लो. प्रिंट आउट लेके उसको रख दो एक बार. Then you can keep on revising almost every 3-4 days once. Okay? Now students, now answer this question, students. Answer this question, all of you. All of you answer this question. Question on the board. See here. Spring. This is the NEET 2024 question. NEET 2024 question. This is spraying sugarcane crop with juvenile. Which of the following plant growth regulators increase the length in the stem and thus increase in the yield? That is basically gibberellins, right? अगर आप शुगर केन में जिबरेलिक एसिड को स्प्रे करोगे और शुगर केन लंबा हो जाएगा. ओके, और क्वेश्चन कैन बी आस्क्ड वेरी सिंपल लाइक दिस. ओके, और समथिंग सिंपल लाइक दिस. ओहो स्टूडेंट्स, एक मिनट, ये क्वेश्चन का आंसर दिया यहां पर. ओके, दिस इज योर, रिमेंबर स्टूडेंट्स, दिस इज ऑक्सिन, राइट? 2,4-D is your synthetic auxin. I told you it helps in weedicide. This is a NEET 2021 question. Okay. उसको यहाँ पर था स्टूडेंट, क्वेश्चन उल्टा हो गया ये तो थोड़ा. Okay, स्टूडेंट, नेक्स्ट इज cytokinins. Now what about cytokinins, students? Cytokinins is very simple. It helps in cell division. अगर आप cytokinins डाल दोगे ना, that entire cell will keep on dividing. Okay, see here. First is chemical nature. They are purines. They are adenine derivative. This also is a PYQ. This also is an NSEJS PYQ also, and even NEET PYQ also. That is, what is the chemical nature of cytokinins? Okay, discovery Skoog and his co-workers, isolation by Miller et al. What is et al, students? Et al are the different other scientists involved in the experiment. Okay, so Miller et al. This you have to remember. Okay, now what experiment did students? Very simple experiment. Skoog and his friends, right? Skoog and his colleagues, right? They were trying to grow tobacco callus. All of you remember what is callus? Remember I told you what is callus, students? Callus is nothing but it is a uncontrolled or group of mass of cells. Generally what happens is when you want to do something called tissue culture. When you want to do something called tissue culture, in tissue culture you take a piece of a plant, leaf or anything. उस explant को आप लेके you keep it on a media, nutritive media. When you keep it on the media, what happens? And along with the hormones there, we get a callus formation. Callus is basically a group of cells, bunch of group of cells, a group of tissues. Okay, so when you kept that different callus here, and we observed that whenever in this callus, right? No, not fertilization. Callus formation. Callus is basically a group of cells. Okay. Now students, here we add the callus here, and we notice that, not we, as in the Skoog noticed that, okay, when you add extract of your vascular tissue, when you add yeast, when you add coconut water or coconut milk, when you add all of these, he noticed that plant was able to grow very well. We noticed that the callus was able to convert to a plant very fast. And later he realized there was one hormone which is common everywhere, that was cytokinins, because cytokinins promote cell division. Okay, cytokinins promote cell division, students. Okay, now students, who did the isolation, students? Isolation was done by CO Miller. Okay, students, so Skoog did the discovery. Discovery is Skoog. Okay, isolation by CO Miller. Okay, and also students, Miller later identified the crystallized the cytokinins promoting active substance, they called them as kinetin. He also observed something called as kinetin. Okay, kinetin, students, kinetin. Okay, this is the kinetin, which is the type of your cytokinins. Okay, substance. And similar to kinetin, there is also called as zeatin. Zeatin is present where? Zeatin is present in the case of your plants. Okay, plants. Okay, so this zeatin and kinetin are type of cytokinins which promote cell division. Now in examination, what they'll ask you? In examination, students, they can ask you questions like this. All of these they can ask you. What are the functions of cytokinins? What are the functions of cytokinins? See, first is what? Regulate cell division. Second is modified apical dominance and promote lateral growth. This is very important. See, auxin, remember students, what will auxin do? Auxin will make sure plant keeps on growing. एक plant लंबा होता रहे, this is done by auxin. Auxin will make sure plant will keep on growing. But what does cytokinins do, students? Cytokinins will make sure plant grows sideways also. इस तरह से plant grows sideways. This is done by cytokinins. It promotes lateral growth. Cytokinins promote the lateral growth. Okay, let's see this. Delay senescence, they'll again, obviously it is a growth promoter, so it will make sure the plant doesn't die. Okay, promote nutrient mobilization, promote chloroplast development, helps produce new leaves and adventitious root shoot formation. So it is a growth promoter in general. Okay, growth promoter in general. Okay, now students, ethylene students, what do I say? It is doing promotion also and growth inhibition also. Dual function is kapura. Dekho, ethylene is a gay, students, very important point. First of all of these hormones till now, auxins, gibberellins, cytokinins, they are liquid in nature, but ethylene is a gaseous hormone. It is a gaseous hormone. Precursor is methionine. Discovery by H.S. Cousins, and bioassay is called as triple response test. Now, how did they discover it? Very simple experiment. Okay, so here we have a ripen orange. बहुत पक गया ये orange, बहुत पका हुआ है. Now what happens is, if you take this ripen orange and you keep it with the raw banana, if you keep this with raw banana, the raw banana will convert into ripen banana very fast. Why? Because this ripen orange is producing some gas. It is producing the ethylene gas. And this ethylene gas can be taken by the banana और ये भी पक जाएगा. और you might have noticed students, अगर आपके घर के अंदर जो apples लेकर आते हो आप, वह banana लेकर आते हो, if you keep them next to the old fruits, जल्दी जल्दी वो पक जाते हैं. Or you keep them under a lot of stress, जल्दी पक जाते हैं वो. Okay, it becomes very ripen very fast. Okay, so gaseous hormone is released, and this hormone is taken by the bananas here. Okay, now students, what are the effects of ethylene on seed and seedlings? एक seed पे इसका effect क्या है? Now you see this. It breaks seed dormancy. Dormancy तोड़ देगा ये ethylene. Initiate germination. उस dormancy को तोड़ के promote seed germination. Now one question arises. Can any one of you tell me which other hormone promotes seed germination? Question will come like this only. Can anyone tell me which is the other hormone which promotes seed germination? That is gibberellins. Gibberellins. Okay. Horizontal growth of the seedling, swelling of embryonal axis, apical hook formation in dicot seedling. Okay. So all of these are effects of seed ethylene on seedlings. Okay. Then they can ask you effect of ethylene on shoot. Again, breaks the bud dormancy. Bud को निकाल देगा बाहर से. It makes the bud grows, right? Sprouting in prototubers. Now this is very important. It will also make sure internode and petiole elongation in deep water rice plants. So in examination, if they tell you increase in internodal length, normally gibberellins is the answer. But internodal length in the case of deep water rice plants, उस वक्त answer क्या है? It is the ethylene in that case. Ethylene in that case, students. Now, in root promotes root growth, promotes root hair formation. Again, very important. Why? Because tell me which hormone, students. Students, PYQ, no. PYQ we will do, no. We will not do this PYQ. First, we have to do the PYQ of biomolecules. After PYQ of biomolecules, then we will do this. Okay, not now. Okay, students. Now, root hair formation affect karega. It will not affect the root formation. Root formation is auxin. Auxin is root formation, but this is root hair formation. Root hair formation. Okay, ethylene. Now, this is the most important one. The effect of ethylene on flower and fruit. Okay. What about flower? Induces flowering in pineapple and mango. Promote female flower in cucumber and hence increases the yield. So again, it has feminizing effect just like auxin. Feminizing effect. Okay. So induce flowering in pineapple and mango. Now what about fruit, students? Now you might have heard, right? Commercially they are used to ripen the fruits. अगर एक fruit पका हुआ नहीं है, you have raw fruit, you add ethylene gas, becomes ripen. Okay, so ripen the fruit after picking. There's something called ethaphon, source of ethylene. So in market you can use ethaphon. Ethaphon can be used to ripen the fruits. It's a source of ethylene. Okay, the plant converts ethaphon to ethylene. Okay, so directly ethaphon can be used. Type of here, right? Commercially used ethaphon, exactly. Commercially used ethaphon is here. Okay. Now students, now what about effect of ethylene on various organs? Normally, generally in other organs, senescence and abscission both. Senescence is becoming older, abscission is decay of a part. Both can be done, students. Okay. So now what about abscisic acid, students? ABA. ABA is also called as, students, abscisic acid. Okay. Chemical nature is carotenoids. Precursor is violocanthin. Right. Discovery is very important because in discovery there were 3 scientists, students. There were 3 scientists there. ये तीनों scientists ना independently they were working. One scientist discovered something called as dormin. One scientist discovered something called as abscissin 2. One scientist discovered something called as inhibitor beta. Later on they get to know all of these 3 chemicals are only one chemical, that is abscisic acid. So abscissin 2, dormin, inhibitor beta, all of these were different discoveries done independently. Okay, but everything is abscisic acid only. Now what are the functions? Abscisic acid is also called as stress hormone. अगर एक plant stress में आ जाएगा ना, it will start producing abscisic acid. For example, students, okay, one second, students, one second. For example, this hormone, let's say increase tolerance to stress. अगर पानी कम हो गया, abscisic acid is produced. अगर पानी कम होने से, it will help in closing the stomata, directly help in closing the stomata. Then general plant growth inhibitor of plant metabolism कम कर देगा. Seed development and maturing, right? Then promote seed dormancy, it will make sure plant dormancy करेगा. Like in the case of gibberellins, promoting seed dormancy था ना. Cytokinins promote seed germination करता था वो, but this is against it. Okay, inhibition of germination होने नहीं देगा ये. Okay, stimulate closure of stomata, or we can just say it is the antagonist to gibberellins. It is completely opposite to gibberellins, like completely opposite to gibberellins. Okay. Now students, I will give you 10 seconds to look at this board. 10 seconds to look at this board. All of it. No, 20 seconds. Time starts now. 20 seconds memory check. 20 seconds memory check, students. And once you're done, you can done sir in the chat. Cytokinins, ethylene, and abscisic acid. So this will be present in the notes. This is the concept of pure memory. You have to remember all this. They have no other option. Students, I told you anti-transparent here. Right? If stomata closes, will there be transpiration? No, it will not happen. Okay. Respiration also does not happen. If the stomata closes, there is no entry of CO2 inside. So respiration also does not happen. Okay. So please remember this, right? Please remember this, right? Sir, again, students, anti-transparent is this only, students. Transpiration will not happen if the stomata closes. It is abscisic acid only. Okay. Now, students, here I've added some questions of all of you, questions for you. So try solving these questions. Okay. They have, see, they can ask a question, match the following question like this. Okay. So, or I've added more questions here. Okay. So this was just a power session, and this one doesn't require much of teaching aspect here. The more effort comes from your side here. So I will upload this PDF in the description, download it today itself, right? And just go through this PDF once. I would recommend all of you take a printout of this. I would recommend all of you take a printout of 1, 2, 3. These 3 slides to remember better. Okay. So thank you so much, students. Bye bye, all of you. Take care and have a great day ahead. I will see you in the next class. Until then, keep studying, keep working hard, and sayonara all of you. Sayonara.